Scientists have a lot of different ways in which we can target molecules at all stages. So from the gene that has the instructions for making a protein to the actual protein itself. Here's just a really quick overview of the different techniques. So the thing that's all in the news, CRISPR. CRISPR is a form of genome editing. You're actually modifying the actual genome, the DNA, and you're doing this in a permanent way. It used to be that you could just do kind of a knockout where you are removing the function of the gene altogether, so you're producing any functional protein from being made. But now you can actually do things like base editing, which allow you to switch a single nucleotide letter, as well as prime editing, which allows you to kind of insert things and do other fancy things. And you can also use like homologous recombination in order to switch regions of the genes in and out. And so you're dealing with the DNA, you're using a guide RNA that's going to target the CRISPR-Cas machinery to this place that you want modified, and it's going to modify the DNA. After the DNA, you've got the pre-mRNA. Here you can deal with splicing. You can use antisense oligonucleotides, which are short modified nucleic acid strands that will bind to things like messenger RNA and pre-messenger RNA in this case. When it binds to the pre-messenger RNA, it can influence the splicing of the molecule in order to make it so that the splicing is corrected in the case of a disease, or you use a backup copy of a different version of the gene or something. So now we've got the mRNA. So the antisense oligonucleotides, those ASOs, can also bind to the mRNA. When they bind to the mRNA, they can prevent translation by blocking the ribosome and other things from binding, and they can trigger degradation by RNase H. RNase H will recognize that there's an RNA-DNA duplex, and it'll cut the RNA, leading to degradation of the RNA. You might sometimes see the word gapmer. Gapmers are where they have chemical modifications on the end of a DNA strand so that RNase H can still recognize the middle, but you've got more stability in your nucleic acid. Also targeting the mRNA is RNA interference, or RNAi. RNAi, here you use short RNAs that get bound by a protein called argonaute. Argonaute goes to the target and causes the mRNA to get degraded. You can introduce these guides with siRNAs, with double-stranded RNAs that get processed in the cells, or with short hairpin RNAs, shRNAs, that are going to be produced by the cell itself and then processed. And they can be produced either transiently in the form of like a plasmid, or they can be integrated into the genome so that the cell will continuously make these, and these are going to allow for a knockdown of the levels of messenger RNA, and therefore a knockdown or decrease in the levels of the protein, but not a knockout like you had with the CRISPR. Now that we're at the level of the protein, what can we do? The common thing is just kind of a drug, an inhibitor that's going to bind to that protein and prevent it from doing its function. You can also use antibodies, and so antibodies are little proteins that will recognize specific molecules. Those antibodies can bind to that specific molecule, say that specific protein you want to target, and then what happens is, depends on what the antibody is, where it's binding. It can prevent binding from other things, or it can actually be some sort of bifunctional antibody where you have it one part that kind of like binds to the thing you want, and then another part that is going to cause some other thing to happen. Okay, so that would be the antibodies. You can also have PROTACs. PROTACs are proteolysis targeting chimeras, and what they have is that they have a molecule that is going to bind to your protein of interest that's linked up to a ubiquitin ligase. The ubiquitin ligase is going to add a ubiquitin tag to your protein. The ubiquitin tag targets the protein for degradation using the cell's natural degradation machinery, the ubiquitin proteasome system. So each of these has its pros and cons, and each of these can be used in different circumstances. Some of them are more for laboratory use, others can actually be used in the clinic, and sometimes the line between the laboratory and the clinic is becoming increasingly blurred as scientists make new discoveries that get applied to help save people and improve their lives. One of the biggest challenges to actually implementing this technology is often delivery. How do you get this to the places that you want it? How do you avoid getting it to the places that you don't want it? How do you keep it soluble? How do you keep the body from metabolizing it and just peeing it out and things? So when you kind of go into the clinic, into people, into organisms, things get a lot more challenging. And so when you do things in a lab and then you try to apply those in a different situation, that's where there can be a disconnect. And so while you might see these incredible sounding findings in the news, but then it never materializes as an actual thing. And then we in the lab can use these tools in order to figure out at the nitty-gritty level how things work, because you never know when that knowledge is going to pay off, that basic research, which is where CRISPR came from.